All right, guys. Welcome back. Over the last few lectures, we've been considering, of course, the TCA cycle, that critical, excuse me, central metabolic pathway through which we can burn things like pyruvate for maximum possible energy yield, right? And we've seen a little bit about how amino acids can feed that pathway, in particular coming through oxaloacetate, um, through oxaloacetate as asparagine and aspartic acid, as one of the being sideways pathways into the TCA cycle through oxaloacetate, or through alpha-ketoglutarate as glutamate and glutamine, and things that can be converted into aspartate, asparagine, glutamate, glutamine, right? Or one of the big ways you can feed that. And there's some other entry points there for bringing amino acids in as well. So, like I said, just about anything that can be converted into an amino acid or into some certain other dicarboxylic acids, or things that can be converted into pyruvate can be burned in the TCA cycle for maximum energy production, right? But the main sort of orientation point we've used really has been glucose, right? This idea of glucose coming down out of glycolysis into pyruvate. And that really is one of the principal under normal non-starvation conditions. That's a major flux, maybe the main flux of the carbon equivalents coming down into the TCA cycle are coming through glucose production, glucose breakdown through glycolysis into pyruvate, right? And then running that through the TCA cycle. So glucose is a big deal. It's a major, under, again, non-starvation conditions, probably the major flux coming down this pathway. So why is glucose such a big deal, right? We started out talking about it. I've really focused my discussions about it, and there's a reason for that, right? Because glucose in organisms like us is, you can think of as sort of a major, or one of the major, one of the two or so major physiologic sort of whole organism energy currencies. It's the way energy equivalents are moved around in our bodies in a big way. So it's worth asking this question: why glucose? What's the big deal about glucose, right? I've made this point before, that it is this really essential fuel. And it sort of bears repeating this point a little bit, that to some extent, I'm not sure anybody really knows why glucose, why physiology locked onto glucose, this particular aldohexose. Seems like there are potentially other possibilities. Could be, this is just a QWERTY phenomenon, like we talked about with L-amino acids, for example, that for some reason glucose was more abundant. The first organisms to evolve a glucose-dependent metabolism or a carbohydrate-dependent metabolism locked onto it, and off we went. Could be there's something about the initial synthesis, or the sort of ease with which one can put together a pathway to break it down is maybe on the sort of evolutionary landscape more efficient. But for whatever reason, glucose is the deal, right? It is the big physiologic energy currency. And it's a big deal really from the point of view of, again, like organisms like us, that there are really important tissues in multicellular organisms like human beings and mammals and many, many others that will only burn glucose, or will preferentially burn it unless they're forced to burn something else under like severe starvation conditions, right? So examples of tissue that are preferentially going to burn are things like skeletal muscle, right? Skeletal muscle in your body, if it possibly can, it's going to use glucose. And only when it runs out of glucose and it's not getting more, will it actually start to break down that protein and then start using it, right, as an energy source, right? But some other examples, right? For example, your brain, your CNS, right? Your blood, I think I've pointed this out before, will only use glucose, especially your red blood cells and your brain. Man, that's all it's going to take. It's not going to accept it. It has no other local storehouses for anything. They're going to keep it running. It really needs to run on glucose. And a major, major point of this really key tissue we're going to talk about, and in the center of regulating all of this is, of course, your liver, right? Okay. So for whatever reason, these tissues, these important key tissues, are locked on for the primary energy input that supplies their function as being glucose, right? And that's not only just for energy, as we've seen, as we've talked about these anaplerotic reactions, these sort of intermediates that are bled out of glycolysis and TCA cycle, it's to provide those intermediates as well, right? And so glucose is that key point, right? Okay, for a lot of these tissues It's the only way to get that physiology, get that sort of energy and intermediate supply going. Okay, so glucose is a big deal, super important. Where do you get it, right? You really sort of have two options as an organism like us. The option number one is to get it from your diet, and that's a big deal, right? One of the main things we're after in our food is sources of glucose, right? Either glucose existing already as starches or carbohydrates in that source, or things that can be turned into glucose in principle. It's one of the big reasons what things we want to extract from our foods, right? But your body's ability to store that glucose is pretty limited. Now, hopefully maybe you remember, long-term energy stores in organisms like us involves fats and fatty acids, right? And fats and fatty acids are a really great place to stick certain amount of energy, because per gram of mass, they will store significantly more energy, several fold, three to five fold is my memory, over say storing carbohydrates. But they're slow to mobilize. Takes more effort to get them to mobilize. And the bigger problem with fats and fatty acids, from the point of view of tissues like brain, right, and blood, is you can't turn fatty acids into glucose. And now we got tissues that can only run on glucose and ketones, right? Lose your brain, lose your blood, you're dead, right? So we've got to be able to make glucose, and there's no way to turn fatty acids. Certainly the fatty acids themselves. The glycerol backbone can be turned into pyruvate, but the fatty acids themselves, as you'll see when you talk about fatty acid oxidation with Dr. Carr here in a few more lectures, are really good for making ATP, but you can't make glucose out of them, right? Okay, so we got a problem. Well, it turns out your body can store glucose, right? And the way it does it is as a polymer like this. You may have heard of it, glycogen, right? We're not going to get to dwell on this much. I just want to kind of mention it here. So it's possible to take glucose, right, and then use the the main sort of connection point to make a polymer of glucose, to make glycogen, to take that 4 alcohol group, use it to attack the anomeric carbon, the carbonyl carbon, and with the dehydration of water, generate a so-called alpha 1,4 link. The alpha refers to the stereochemistry about this bond. You can also have a beta, but the alpha is what's relevant for glycogen. And you can just connect glucose molecules, these alpha 1,4 links. And when they get to a certain length, 10 or 12 sort of residues in length, there's another enzyme that'll come in and make a branch point, so-called alpha 1,6 branch point. Same basic idea, just uses the 6 alcohol instead of the 4 alcohol group. And that'll branch off, and then it'll go 10, it'll branch off. And what this builds, there's these big branching dendrimer-like spherical structures called a glycogen granule, right? There's a little protein called glycogenin. That's a little tyrosine residue on its surface. That's the nucleation point that gets one of these chains going. And they build these dense, super dense little granules. And here's an example of some liver tissue. And those little black spots are nitrogen granules, right? So this is the way in which your body, your liver and your skeletal muscle, your muscle tissue, does this as well, stores lots of glycogen granules. But the problem is that the metabolic load for an organism like us, in terms of glucose flux for those tissues that really will primarily use or will only use glucose, is enormous, right? Average human being uses about 160 grams of glucose per day. And about 3/4 of that, about 100 to 120 grams per day, is necessary just to keep your brain and your central nervous system running, right? You lose your brain, you're gone, right? No more breathing, no more autonomic response, you're gone, right? So big priority, keeping glucose going into the brain and the blood tissue, right? The problem here is that your liver's ability to store this glycogen is limited, right? Your liver can store about a half to a day, 12 to 24 hours of glucose supply in these glycogen granules under starvation conditions, if you're not bringing in any more. So that's about it, about a day, and then you're out of glucose, right? So then what do? You got about a day, right? That's all your stores are. And it may be hard to understand in this, current point of human development society, but in the evolutionary history of our species, it was anything but normal to get an adequate daily supply of glucose, actual glucose, from your diet. It was sort of a windfall kind of thing to find a good source of readily available sugar like that. I know again, it's hard to imagine with Red Bull and candy bars and whatever else in the modern diet, junk food. But this was not the thing that our species, through most of its evolutionary history, and our um, pre-hominid ancestors faced. They had to find other ways to get this in, and there are certainly other things that one could use to make glucose, right? Because remember, we've got other ways to bring carbon equivalents into the TCA cycle, right? And it turns out by using one key intermediate in the TCA cycle that we can connect up to things like pyruvate, right? Or other amino acids. Let me just write that out, right? Or lactate, we're going to see an example of that, right? Some of the ribose and deoxyribose sugars. There are ways in which, there's a way, really important way, in which dietary sources of things like pyruvate, protein, amino acids, lactate, deoxyribose sugars from diet, these can be turned into glucose, right? So I said there are two ways you can get an adequate supply of glucose to keep those critically glucose-dependent tissues running. Diet, right, where you just get the glucose straight from your diet. The other possibility is to take these other sources, right, of carbon equivalents, right, and figure out a way to make glucose out of them, right? And that's the process we're now about to start thinking about. It's the one anabolic pathway we're actually going to spend a little time dwelling on because it's important for understanding how this whole process of glucose metabolism is regulated, right? And this is, of course, a process known as gluconeogenesis. Oops, misspelled it. Or literally the synthesis of new glucose. Okay, and we're going to start in with an overview of that next video.